Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we'll be talking to Emmarie Alexander and Tyler Chan. Emmarie and Tyler are in their third year in the interdisciplinary graduate program in genetics here at A&M. Today we will talk about their third year as a graduate student and what life is like after passing their prelim exams. My name is Sam, and I'll be one of your co-hosts today.
And my name is Veronika, and I'll be your other host today. Welcome, guys.
Thank you.
Welcome to Blue Jeans and Boots. I think first I'm just gonna go ahead and ask you, how has your third year in the gene program been so far?
Oh. Tyler, do you want to start or do you want me to start?
She threw me under the bus first.
Well, actually, wait. Before we do that, should Tyler and I introduce ourselves? Maybe give a little context on who we are if you guys don't know us.
Yeah, only if you go first.
Let's do it.
Okay,
please.
I will go first this time.
Go
Okay.
ahead.
Howdy, everyone. My name is Emmarie. I'm a third-year PhD candidate. Whoop. I'm in Dr. Bill Murphy's lab. Our lab uses comparative genomics to study adaptation, diversification, and the evolution of mammals. In particular, I study hybrid male sterility and sex chromosomes across a bunch of mammals, but my particular focus is on cats.
Very nice. I'm Tyler. I am a third year, also PhD candidate in Dr. Zachary Adelman's lab. I work mostly with genetic control methods for arthropods, so a little bit of mosquito work, namely Aedes aegypti, the yellow fever mosquito. But sort of my primary focus is on biting flies, and so I do a lot of microinjections, transgenesis, and genome editing.
So both of you mentioned that you're PhD candidates.
Yeah.
Mm-hmm.
So for those who are not familiar with how graduate school works, can you tell us the difference between a PhD student and then a PhD candidate, please?
For sure. So oftentimes it's really dependent on the program and the department. For the genetics program, you come in as a PhD student, and then about at the end of your second year, the start of your third year, they will ask you to undergo this process of qualifying in order to become a candidate for your doctoral degree. And so we'll talk a little bit more about the process, but this oftentimes involves writing a proposal about what your dissertation is going to focus on, in addition to either writing a grant, depending on what department is your home department and what their requirements are, or doing written or oral exams, or sometimes even both. So yeah, you come into grad school as a student, then you become a candidate, and then you become a doctor, which is still a little bit out for us. So yeah.
Yeah, I've heard it called qualifying exams, preliminary exams. Our program, the gene program, has pretty loose requirements. You just need to submit a project proposal, do some kind of routine paperwork, but most of the prelims are kind of set by your PI and your committee, so it varies a ton, just student by student. But the general idea is that it's sort of like a pretty big halfway hurdle to make sure that you have the critical thinking and reasoning skills to make it to the end of your PhD and be a full-fledged doctor. So it's really kind of evaluating your capability to finish your degree program.
Yeah.
So you mentioned committee. So what was your process for your committee choosing like, and what was that experience for you?
Sure. Do you want to go first this time?
Sure. To be honest, the field of pest arthropod genome editing at Texas A&M, the pool is actually smaller than you'd think. So there's kind of a handful of candidates for a lot of reasons. When you're choosing a committee, you really want people that are kind of not just giving you advice for your project, but also kind of like they're the people who are going to have your back because they decide if you graduate with a degree or if you don't. So, a lot of times people, PIs vary a lot, but if your primary investigator says, hey, I think you should choose these people, just keep in mind that you have total liberty to choose whoever's on your committee. However, I will say Dr. Adelman gave me pretty good advice on who to pick for my committee, and they're all very, very supportive, and they're kind of a mix of, people who I like their mentorship styles, but didn't really do anything in my field, and people who are very much in my field, asked really good critical questions, but are more like subject matter experts than they are just like support members. So I like to have kind of a mix of people on my committee.
Yeah. I kind of think that I won the lottery in terms of grad school and also my committee. So if you guys aren't familiar with our genetics program and how it's structured, as a first-year student during your first semester, you'll have to do rotations, and oftentimes you'll do 3 rotations, sometimes a 4th. And all 3 people that I rotated with, I just got along with so well. Their research interests really were aligned with what I wanted to do, and I also just really appreciated their mentoring style. I'm going to give some shout outs. So Jessica Light, Keith Blackman, those were 2 people that I rotated with, and then also my PI, Bill Murphy. And yeah, I just think that they're all like not only just outstanding scientists, but also just outstanding people. And I don't know, I feel very well supported by them, and I feel like whenever I have a question, I can ask them and they will, poke a hole in my research, but it's always for the best. And then the other person that is also on my committee is Terry Rodsep. So as I had mentioned, I study like comparative genomics, so oftentimes I'm looking at chromosome structure and chromosome evolution, and Terry is like the most phenomenal animal cytogeneticist. She's a rock star. So like I said, I feel like I really won the lottery because not only do I have like really outstanding scientists that work on sex chromosomes and mammal evolution and phylogenetics, but they're also just like such great people, and it's really like my committee meetings are fun and I look forward to them, which is not something most grad students can say. Yeah. So that was kind of what that process was like. Yeah.
That's amazing. Thanks for sharing that. So can you remind us, please, when did you do your preliminary exams?
Yeah. So mine was the second. So, okay. I'm going to talk a little bit about structure too, because this is important. So the second week in November, I had a written exam per day for each of my committee members. So that was 4 written exams over the course of a week, and then I had Friday off. And then the following Wednesday, I had my oral presentation or exam, which was about 2.5 to 3 hours. Yeah, so it was most of my November was either stuffing myself with food during Thanksgiving and fall break, or taking exams. So I say that was with hesitancy because it is kind of really, really, really stressful. Yeah. Anyways.
Mm-hmm.
Yep. Yeah. Honestly, mine was pretty similar. Unfortunately, I didn't have the joy of Thanksgiving to keep me warm during that time.
I'm so sorry.
It's okay. It was just a rather dreary October, but it was honestly. I have 4 members on my committee, including my PI. And one of them, Dr. Ching Sun, she elected not to give me a written exam, so that was kind of a nice reprieve. But my other 3 committee members gave me written exams. Dr. Adelman, he said, you have 24 hours to do this, and he just let me choose any dates before my prelim to get that done. One of them was just, you have sort of a week to finish it, Dr. Emmarie. And then Dr. Kevin Miles just said, as long as you turn it in a reasonable time period, he said, I'll be happy. So all those written exams I kind of scheduled a good couple weeks before my actual oral prelim defense, and it was all kind of some of them were questions that were a little bit more out there, some of them were very subject matter oriented, but basically I just sent a lot of back and forth emails to my committee to ask them questions, send back my eventual responses to the written portion, and then kind of get some studying feedback before the actual day of prelims.
Yeah, I should probably add that the way that I approached it was pretty unconventional, and we didn't really catch it until it was pretty close to the date. Usually you want more of a break in between your writtens and your oral exam, mostly so you can do what Tyler had mentioned, where you can review it, you can receive feedback from your committee. But when I had brought it up to my committee, they were like, we're not really worried that much. Just take your exams and we'll have a good chat during your orals. So it's really dependent. Yeah.
So I guess you kind of started talking about it, but how exactly did you prepare for your prelim exams, and what was that like?
Yeah. Okay. So as Tyler had mentioned, one of the main requirements for candidacy in our program is that you have to submit a dissertation proposal. And this proposal is usually 10 to 15 pages long, and it sort of breaks up each of the chapters of your dissertation, and you provide an intro, some of the methods, and your what you kind of expect are going to be the results and implications of that research. And I think that is a really good study guide, especially for those more like study specific questions where they like really want to get like nitty gritty about the science. The other way that I prepared is I asked my committee members if they could send me papers or at least tell me topics that they were going to test me on, but they all very broadly were like evolution, sex chromosomes, mammals, like know it. And I was like, okay, I will. So from that though, I sort of thought about a lot of foundational literature in those topics. And so I would kind of structure my day. I would try to read a paper a day during lunch. And then oftentimes, I spent a lot of time on my proposal, but that was also because I was in the process of like prepping to write a manuscript as well. And so I blocked out a lot of time every morning to sort of write, read, and then spend the rest of my day doing science. And I did that starting in August and then sort of slowed down by the end of October and then gave myself a sort of grace period where I just like didn't do anything and just like tried to have fun before I had to take really hard exams. So.
Wow. That's really thought out and like well organized. I can't say
I'm
that I did the
super
same
anal.
thing.
What can I say?
Yeah, that's a great quality to have, especially for research. To be honest, I feel like my studying kind of started on like the year before, because the other people in my lab were taking prelims. Well, I guess that would have been 2 years ago. Yeah, so I've seen people in my lab kind of each year get ready for the whole prelim process, and I actually just kind of sat in with them and watched their prelim studies, and that was a really informative process. So if you're like a first-year PhD student, maybe talk to other people in your program that are like a year ahead of you and just watch them get ready for prelims. It can be pretty insightful and pretty good practice, especially if they're in your fields too. So the way that I saw a lot of people practicing is they would just, find someone who's, in a somewhat related field. You just write down a ton of questions of like things to review that night and go to a study room in the library. And you just have someone explain like one of their thesis aims and the other person just like ask some questions until you get to the I don't know moment. And also, they would just kind of say like at any point in time, they would say like, explain this acronym. What does this mean? Can you draw this out on the whiteboard? And my prelim ended up being pretty much exactly like that. So I think that format for a lot of PhD students is a pretty good way to study. So
Yeah, I think that's pretty great. Another sort of like pro tip is oftentimes your committee members will expect you to have taken their classes and they'll ask you content that they taught during their class if you were a student. And so that happened with my own advisor. He like followed up with like some of the content that he taught in his class. And then that also happened with another person on my committee. She asked me questions about content that she taught that was relevant to my dissertation.
So you mentioned that you had both an oral portion and also a written portion. And it sounds like the written portion, it really depends on the PI or like on the PI that's on your committee and how long do they want it back or to be submitted, which is quite interesting in my opinion because for one PI you need to do it in 24 hours, but then for the next one in a week, right?
Yeah. I would say that actually that is one of the things that I found really stressful about prelims is that it is not a standardized process and that you really have to have open communication with your committee members to like fully be aware of what you're like submitting yourself to. So yeah, I had 2 8-hour exams. No, I 3 8-hour exams, and then I also had a 24-hour exam. And like some would give me questions where I could like they would give me 5 questions and I could pick 4 to answer. Some of them where I had to answer every question. One of them was I had to perform like basically a critical analysis of a paper, 2 papers, and compare them and their findings, and then also write a mini grant proposal. So like it was kind of all over the place. And yeah, you usually can ask your committee members how they plan to structure it, but just know that oftentimes they don't even know until they sit down and write the exam.
yeah, keep in mind your committee is also very busy, and if you ask them like, what are you going to quiz me about? Usually that they might not know until maybe the week of or something along those lines. So yeah, keep in mind the human element. Another thing too is what you said about open communication with your committee. When you have those committee meetings, I know our program, mandates that we have like one committee meeting per year, just to keep in touch with your committee and all that good stuff. That's a really good time to gather everyone in one room and say, hey, I've got this upcoming prelim. Tell me, what time, what months work best, what weeks work best, what are you going to quiz me on? Do you like written exams? Do you hate them? What do you expect? Like, what do you see in like an optimal candidate performance? I think those are like good starting questions, and just follow the thread from there, and keep following up with emails, because sometimes it can be hard to keep in touch. But it's good to know kind of going into an exam, because if someone says like, we're going to quiz you a ton on let's see, microRNA or RNA interference, you should probably read your committee members' papers on that subject, because that'll give you a really good idea of how.
Definitely. The one other thing too that I just want to remind people who are listening that are in graduate school is that your dissertation is going to evolve over the course of 5 to 6 years, and the things that you initially state in your first committee meeting are probably, they might be the same things that you're going to be working on by the time you take your prelims, but also they might not be. And so I think that you need to kind of remind your committee members of what it is that you do and what it is that's going to be relevant for them to ask you. But they should also still have a pretty good gauge on that, because you asked them to be on your committee for a reason. So
I think that was pretty great advice, because especially both me and Sam are second years at the moment, so we just received that email about thinking about prelim dates and scheduling our second committee meeting to even debate that topic. So all of this advice was actually very helpful, I think, for us too. And I think it's interesting, because when you come to a PhD program, you don't really think about prelims your first year. You're just happy you got into a grad school, happy that you can do finally research in the lab by yourself eventually. So even thinking about this topic I think is pretty important from the beginning, because you don't know what the format is until you start asking questions, and you realize that it's definitely not the same for everyone, and it's very person dependent.
Yeah, and I think on that note, also, please be kind to yourself when you are scheduling prelims. I really like the way my committee actually told me that they said that I could really take it whenever in fall, but they were like, there's a really good chance that you're going to get burnt out and you're going to be just frustrated and you're going to be overwhelmed, and it's a very normal thing, and that you should give yourself grace, and that if you can take a break during fall break, and so you have, you work up towards this point and you have this big thing that is really important and you pass and it's a great time, and then you can also just take, a couple days to step back, to reflect, and just sort of I don't know. Grieve isn't the right word, but sort of just process everything that just happened over the couple of months. And so I don't know. I think it's two things. Don't feel super obligated to rush into your prelims. If you don't feel ready, there's nothing besides, the gene program asking you to do it by your third year. But do it when you're ready, and then also know too that your committee members are not going to say yes to you taking your prelims unless they feel that you're ready, or you just have people on your committee who are mean, which hopefully doesn't happen. But your advisor first and foremost shouldn't agree to you taking your prelims unless they feel like you're at a point where you're going to do okay. So
So how do you feel now that you've finished all of that and you finished your prelims a couple months ago? How do you feel?
Boy, oh boy.
Yeah, that was a funny response. Honestly, I think the day after, it hadn't really hit me yet, because it was just, a lot of studying, and I tried to get a head start so I wouldn't, be just dissolving in this pool of literature, and at some point you'll start seeing papers in your dreams, and at that point it's time to take maybe a Saturday off and go for a run or something. But so day after, went into the lab, worked normally, and then just like I think the day after that I slept for like 13 or 14 hours, and then I just kind of said, you know what, I think I needed that. So honestly, it feels really good to be done with prelims. I'm also done with teaching, so right now I'm very much a monotasker. I like to just kind of focus on my thing and plow ahead with it. So it's, I think, much less stressful now that it's done, and also you have a much more predictable schedule. There's fewer like crazy looming deadlines, and you can just kind of like check off boxes and check off progress. So
Yeah, definitely. I've been told by many people, professors and grad students alike, that prelims are going to be most likely the hardest part of your PhD. And so far, I mean, I'm not all the way done, but I would definitely say that prelims have, like, they were hard. I think it's also hard because it's like, you recognize that, like, you can do it, but there's going to be moments where you feel like you can't, at least for most people. And I think it's, like, a lot of just, like, convincing yourself to, like, do it and that it, like, you gotta get through it. But it is really, really stressful. 'Cause you also kind of feel like, I don't know, like if you don't pass, what happens then? Like, there's what ifs. I think all scientists sort of, are looking at the null and the alternative hypothesis, right? But yeah, I feel a lot better now that prelims are over. I feel much more confident in myself as a scientist. I feel really excited about science and the fact, like Tyler said, I just get to sit down and do research and grind it out, and like I'm happy. And that's not to say that like I didn't enjoy studying, because like I can bust out like literature and citations. Like I'm like, okay, but do you remember in like Makova et al. 2024? Like I can do that, but I don't know. Like I wanted to be a grad or like PhD student so that I could do research, and now I can like do research.
Now you can finally do it.
Yeah, and
Exactly.
so yeah,
That's amazing.
Yep, that's a good feeling.
it is a good feeling. Yeah.
One like side note on that, you said like what the alternative to passing your prelims. If anyone out there is scared about like what happens if you don't pass them, one of the best scientists in our lab right now, he's fantastically skilled. Didn't pass his prelims on the first time, and he is, of almost intimidating, if not downright scary, intellect and just field knowledge, and like really, if anyone's gonna ask you a really piercing question during a lab meeting, it's him. And if there's a lot of people out there like that who did not pass their prelims on the first try, so keep in mind, even in the worst case scenario, you're still gonna be okay. So,
Yeah.
So in those moments where you were burnt out or really overwhelmed or stressed out, how did you handle that, and what was your kind of go-to cure is not the right word, right? Because it's not going to be cured, but remedy or activity,
I think doing stuff that like kind of made me just feel grounded and like kind of took me a step away from science, because like obviously like we love science, that's why we're here, but in that moment science sort of feels like it's everything. And so I really like to be active. I love weightlifting, I love rock climbing, I love being outdoors. And so when the moment arose, I would try and go to the crag and climb outdoors. Or like travel to a different gym with some of my friends. There's actually photos of me that some of my friends have where I have textbooks in my laptop, and at like between breaks of rock climbing I would actually be studying. But like, I just needed to stay active. And also I think another thing too is that like you need brain breaks. Like you really need to like just kind of like rot on your couch and like not do anything. That's like really mentally taxing, because like studying is a lot of mental effort and energy.
Yeah, I'm gonna double down on that. Lifting weights, best exercise. Don't quote me on this. There's a paper out there that I've seen that has, I think it was deadlifting in particular was like more effective than a placebo in reducing depression symptoms or something like that. So lift heavy weights, make the sad voice go away.
Yeah.
But yeah, honestly, I did a lot of the same stuff during prelims. It's like you probably work pretty hard all day in the lab. You come home, you study, you wake up and you study. Take a few minutes out of your day just to get some fresh fruits and vegetables. Make sure you're getting 7 hours of sleep-ish per night, and Yeah, I think honestly being physically active kept me pretty sane during that time. I joined a sports club and just hang out with some people, do something quasi-social, quasi-exercise. I think that's really important. Sometimes it can feel like you really need to study that night, and like, obviously it is important to really just get that book knowledge in, but at some point you got to make sure that what you're doing is sustainable. So hang out with friends, balance things even when you are working hard, and I think that'll probably result in much better prelim day.
Definitely. I guess kind of along the same lines, how did you kind of practice having grace with yourself?
Yeah.
Because I know for a lot of people, and for me, that's a very hard thing to do. If you were successful at it, by all means, please share your, tips and tricks.
Yeah. I think it's mostly just like having some sort of like self-affirmations. Also like silly quotes and stuff like that. Like I have hanging up around my office and like around my desk, and like ironically, Darwin's birthday was yesterday, and Darwin has some pretty great quotes of him calling himself stupid and moody and grumpy and like that he hates everything. And like, me too, Darwin. you know? And so, yeah, I think it's kind of just like recognizing that like that sort of stuff. I think also just like what Tyler said, like, if you really feel burnt out that day, you have to like recognize that like no amount of studying is gonna like improve that. How do you practice having grace with yourself, Tyler?
I'll take it from there. Honestly, I really don't, to be honest.
Oh, no.
So this is one of the things. I think there are millions of PhD students out there right now and researchers, and there's an equal number of ways to do a PhD. I don't really do like self-affirmations. I just kind of wake up, I'm like, make the little checklist, and if it gets done, awesome, go to the gym. If not, self-flagellation, 1,000 whips. Just kidding. But I think it's important to know what motivation style works best for you. That's how I'm going to spend this. But to me, I think sometimes I do have to look in the mirror and be like, dang, eating 10 ice cream sandwiches and not studying is not as good as probably sitting down right now and cracking open those 3 papers that I should have read a month ago but didn't read.
Yeah,
So
that's fair. I will say though, sweet treats go a long ways. Like if
So
you
true.
can condition yourself into studying,
I do that too.
yeah,
Mm-hmm.
it's pretty bad. Just don't get to the point where like you get a sweet treat and you don't do anything.
Oh yeah.
I do that with chores.
Yeah,
Especially with folding laundry. I'm like, if I fold this, I can have a bit of ice cream.
Yeah.
Ooh.
I do that.
I use cleaning to procrastinate too. So
Yeah,
sometimes
same. Productive procrastination.
So your
goes
houses
hard.
were nice, huh?
Oh, during prelims or getting ready for prelims, it was spotless. I mopped the floors. It was.
Yeah.
What would you say was the biggest challenge during this whole preparation process?
Sorry, I feel like a lot of my answers have been sad. So I would just like to say sorry about that. I think honestly, I was like really scared that I was gonna fail, and like I think going back to your question, Sam, about like having grace with yourself, like I had to keep reminding myself that like even if I failed, it wasn't gonna be the end of the world, and like also it like wasn't gonna define who I was as a person or as a scientist. But there was like a lot of fear of just failing and kind of being like a disappointment to like my lab mates and my advisor and like myself. And yeah, like that was really challenging for me because I was like, what if I like try my hardest and it's still not enough? Sorry, that seems really sad. But it's kind of the truth.
But you came through.
I came through. Yeah.
Yeah, and it all worked out.
Yeah.
There we go. What was the phrasing of the question was, what was the hardest
Yeah, what was
part?
like the biggest challenge you faced in your preparation?
Honestly, I think the preparation, since I started like really early, I think it was probably just keeping myself on a tight schedule. But I think also my life was just pretty busy in October. So balancing, both just like, life outside of work and prelim prep, there's only like so many hours in a day. And when you have to do like 25 hours of work in 24, it's, it's challenging. I think there was a lot of luck. I think there was a lot of, support from friends and family. So like, on Sundays, call my family, and then having that social support network, I think really helped. But yeah, just balancing the hours in the day was probably the hardest part.
I would like to clarify that my lab and my advisor are super, super, super supportive and always tell me that I'm not a disappointment and that I'm a rock star. So that was a lot of just me being sad, being a sad girl.
Well, it's a tough topic, just prelims by themselves. So obviously we're not like, yay. So
yeah, imposter syndrome's real. So
it's a hard exam.
Yeah.
So it makes sense that we're a bit stressed about it.
Yeah.
So what kind of advice would you have for us two, for example? Because we are kind of getting there slowly. Yeah, we are. Yeah, we are. We are.
I think maybe like Tyler had mentioned, identifying things that like you can do that are going to keep you sane. So whether it's being active or like trying out a new hobby, reaching out to your friends and family and also your lab mates and your advisor, having open dialogue about just how things are going and like checking in with yourself and checking in with other people. Yeah, I would say too that like some of my friends and also like my boyfriend, when I was taking prelims, during the week of my writtens and then also like my oral exam, they like cooked food for me and like came over to spend time with me after I finished and like brought me coffee. I just dropped it off when I was taking the exams and just like checked in. And I think that like that amount of support really like got me through it, and it felt really nice to be like knowing that like, you know, I had a village behind me trying to lift me up. So yeah.
Sorry, so you said advice for you guys. This is, I think, not nearly as like kind and sentimental as that. So this is just very like dry and technical, but I think one of the best things I did was scheduling like I meet with my PI like once a week just to give like a quick lab update on Fridays. Yeah, like 30 minutes, you know. I don't know if your all's labs do that.
I do that.
Okay. I would probably ask him if he can like get on his calendar and just say like, hey, can we do like some prelim prep sessions? I think knowing the kind of questions Dr. Adelman asks some very, very good and insightful prelim prep questions, and it kind of gives you like an idea of like not just the difficulty but also like the subject matter that might be asked on in a prelim. I think those were maybe the best studying that I got, and doing those wasn't just beneficial for the prep, but it also made me feel a lot more confident, I think, going to the exam. So I think familiarity is confidence, and having something just on the calendar where you meet every Friday, every other Friday, like in the 2 or 3 months preceding, I think that gives you a really, really great foundation going to prelims.
Yeah, that's a great point. Okay, can I ask you a question, Tyler?
Shoot.
Okay, during your oral exam, did you find that when a committee member had asked a question, like that even though you knew the answer, you like would forget the answer?
I think there was one point where they actually had to tell me to like stop talking, because I was really excited to share a lot of my prelim research.
Yeah.
So I would actually share too much, and they would say, okay, like we gotta keep going. But I think there was a couple times where someone asked me a question, and it was something that I'd studied so many times, and then I just forgot the name of a protein, and I kind of looked like an idiot. But I eventually, you know, I would say like, you know, pardon me, like give me like 5 or 10 seconds.
Yeah.
And your committee will usually be nice enough where, you know, they'll allow you to take a minute or two, just recover, think about it, and then you can kind of go on.
Yeah.
So that happened to me like once or twice.
Yeah, oftentimes they'll ask like guiding questions too if they like know that like you know, but you're having like a hard time like arriving to that point. I will say though that it is pretty common. It happened to me and it happened to a couple of my other friends, not in genetics, but like in biology, for example, who were taking their exams. They would like say something that was literally on their PowerPoint slide, and then like their committee member would like basically ask like a really easy clarification question, like basically reiterating what was on the slide, and then they would be like, and they were like, no, you know, like you just said it came out of your mouth. Yeah. Also, there's gonna be questions too. I think this is like a good thing to know. There's going to be questions that your committee members ask that they don't know the answers to. If you have the right committee members, they're going to be really excited about science, and especially your science, and they're going to have a lot of questions about it and just questions that they're curious to. Yeah, my own PI, like after he came up to me and he was like, Emmarie, I just want you to know that there were some questions that I asked you that were really hard and I didn't know the answer either. But yeah, so.
Yeah. Honestly, prelims, they're pretty much your committee is going to ask you as many questions as they need to find out where exactly you don't know. So you will eventually say, I don't know. That's the extent of my knowledge, or like, that's a boundary. You can make an educated guess as long as you're very clear that like, this is some speculation, but yeah, you will be quizzed to the full extent of your knowledge.
Yeah.
Also, I had when I was doing my practice sessions with my PI, you really have to be prepared to answer anything.
Yeah.
I walked in on a Friday and Dr. Adelman said, ants, what do you know about them? And I said, well, some things. And he's like, and he just started talking about a paper that came out recently that found that leafcutter ants, which, kind of farm fungi, they did a big phylogenetic study, and he's kind of asked me to reason through it and also try to guess why he was asking these questions. And the end result was that they found out that all these different species of fungus that they farm and all these different leafcutter ant species kind of originated, it is thought at one point, and that was the KPG extinction.
Cool.
So they think that basically there's tons of decaying plant matter and all these ants just grabbed leaves and went underground. But, I came in expecting a normal prelim prep session, and I got, I started hearing things like, what do you know about the Cenozoic and the Permian and the Triassic? And I was like, so, there's a lot of, depending on your PI, you might have to do a lot of really, really like outside your comfort zone reasoning.
Yeah.
So practice that with your friends too.
Yeah. Oh, okay. Actually, one more piece of advice. Read the dissertations that other students in your lab have written. Yeah. Especially ones that pertain to your research. Yeah.
So I know that this is going to be very specific and personal to each person, but how broad do you recommend going with the literature reading?
Yeah.
Kind of what you were just saying about, he was quizzing you on ants, but yeah, I think it's hard to know exactly how far to go, but
Yeah, there were definitely a few topics, or not topics, but I would say sort of papers that I really honed in on, and then had no relevancy at all to what I ended up getting asked. I think it's also, it's very dependent on your committee members. As I mentioned earlier on, I study speciation and how different mammals are related to each other. And almost every single one of my committee members asked me what a species was, which seems really silly because, I'm sure everyone, well, this is the problem. Everyone has their own definition of what a species is. So what does someone who studies speciation think it is? So I thought that they were going to ask me about certain genes and proteins and parts of the genome, and no, they asked me what is a species. So I think just knowing that you're going to have some weird questions and things are going to be broad, but I guess kind of try to pick out a few main points of your dissertation and the overlap between your committee members in your research, and maybe focus on that.
Yeah, in terms of how deep and broad you need to study, I think that kind of depends. A lot of my prelims were very concept level, but eventually it did dive down into, what is this very, very specific pathway, and how does it work in these mosquito species? How does it work in Drosophila melanogaster? How does it work in Drosophila, Suzuki, Heidii? How does it work in your fly? And I kind of built, remember in elementary school where you had to draw the concept maps and just lots of little bubbles and lines? I had this really horrifying one to look at, and it was kind of what I gathered during all my practice sessions, and it was just written on this yellow legal pad, and it looks like a crazy the Always Sunny in Philadelphia thing.
Yeah, Charlie pointing at
It looked like that, but that was all the concepts coming off of my 3 thesis aims, and every question I've been asked about all of those, it kind of gave me an idea of what to hone in on. I didn't really do much RNAi interference, but one of my committee members, Dr. Miles, works a lot on it, and he has a background in virology too. So I really had to learn a lot of stuff that was outside my comfort zone, just because a lot of the questions that my fellow lab members, who also had him on their committee, they got a lot of questions from him, so they asked those questions to me, and that helped me kind of round out this like kind of like a nebulous space of ideas and questions. And I had to find a lot of like specific papers to get answers to the questions that had been asked during my studying. And I think that probably gave me like 95% of the information that I needed for prelims.
Would you say that your writtens were like really broad, conceptual, and then the nitty gritty happened during the oral? Because that's kind of how I feel. Is like there were a couple things that I felt were like really, yeah, like specific for my writtens, but most of it was like broad.
Oh, wow. One of my writtens was very much just like kind of describing a pathway in some species. So
Okay.
I think that kind of falls under the specific
Yeah.
area. Dr. Adelman's question was kind of like comparing and contrasting two similar papers that like simultaneously discovered a similar protein in like at the same time. So that was like kind of like a meta, analysis. It was more like, comparing and contrasting different methods, approaches, and theories that still got to the same result at just about the same time. And that was, I don't know if you'd call it like specific or more like, I guess it's more specific.
I think it's both, right? Because you're being asked to critically review or like analyze scientific like results, but I think it's also specific at the same time. So
Yeah. So I think mine were probably more specific than yours.
Okay.
But honestly, my prelims are very like open-ended.
yeah.
How did you organize the material that you used to study, or like the literature that you were focused on, anything? Like, how did you have it together?
Yeah, so PaperPile, if you're listening, I need you to sponsor me. I advocate for your software so much. PaperPile is like the GOAT of citation managers. I really, really like it. It like I'm able to organize it, highlight it, like send it. We have like shared lab folders where everyone can like compile papers that they want other people to read. So I basically have like, evolution, sex chromosomes, mammals, and then I break it down into like mammals is categorized by like all the different species that I work on, and then like sex chromosomes are like all the papers on the X, all the papers on the Y, and then like genes, recombination rates, phylogenetics. And so like I would have like basically like a main category and then like a bunch of subfolders, and then I would also try to take notes and like sort of like summarize the papers that I often read, because that I feel like is the best way to sort of remember the contents of them. But I all did that, like I did the notes in OneNote, which is where I house as a bioinformatician, I do mostly dry lab, so all like my lab notebook and basically my whole PhD is in that OneNote, so including my preliminary notes.
Oh my gosh, that's terrifying. Do you have like a million backups of everything?
Yeah.
Okay, that's good. I'm really bad about backing stuff up. So I can tell you how I did organize it or how I should have organized it.
Please do both.
Okay. Well, Dr. Noriko Inaguchi from University of Alabama in Huntsville showed me the most immaculately organized PhD. She, like, every component of her PhD, she's a postdoc now, was in this, like, 3-ring binder, and it had, like, every document ever, all the revisions and iterations, and it was all, like, tabbed and, like, color coordinated, and you flip through it, and it's just, like, it's almost like a first-person narrative where it shows you like the exact flow of how like when her ideas changed, what feedback she got from her committee, and that's how I tried to do it, and I did not succeed. Honestly, Zotero, if you're out there, the superior reference manager, you should sponsor me. They probably don't have any money since it's like free and open source, but I love Zotero so much. But yeah, I have all my papers on there. You can kind of add notes in the sidebar, and that's what I really did for a lot of them. So it's just all the notes are stored as like open text files. You can highlight everything. All the PDFs are stored locally, so you don't have to like worry about cloud storage or like acquisitions. I know Mendeley got acquired by someone a while back. I don't know the details, but yeah. So I had this horrible mixture of physical notes, digital notes, an Obsidian notebook. If anyone takes notes in like Markdown, and I got really enthusiastic about learning Markdown, and then I just realized that I really hated looking at my computer more than I had to. So then I had like 4 or 5 competing note systems, and I eventually just went back to writing everything down in a like college ruled notebook, and then doing all of my paper reference stuff in Zotero. So I recommend any like first year PhD students or even, undergrads that are like really into research, get a citation manager and, stick with it.
Yeah, I think consistency is really the big thing about like staying on top of stuff. Like if you try to like take on like a new thing, like I know a lot of people, oh my gosh, what's that app? One that you can like make like, I don't know. Notion. Yeah, so many people are like on the Notion train. Yeah. Yeah, exactly.
Yeah,
like I think that a lot of people like they start out because they have like they're really like bright and spry and they like want to do a good job at studying, so they like want to pick up a new habit and like use a new like note taker, and like things are going to go good, and things usually like don't go good. Like just do something that you feel good and that is going to work for you.
Well, and as far as the like citation management goes, I tried Notion.
Yeah.
Because they have like people can go in and they have their templates or whatever, and it just was not the vibe for me.
Yeah.
I don't know. It was very hard to organize my thoughts appropriately. So I don't know. Maybe y'all can skip Notion. That's maybe not the vibe for that. Yeah.
Yeah.
I have too many hand-drawn scribbles to use. I bet an iPad would be nice because I've seen people like take lots of like really good notes in I don't know what software it is, but
I take most of my notes on
Okay.
iPad, and I download a lot of papers
that's
on,
cool.
and because I can highlight directly on there, and like take notes physically, because I definitely am like, I have to like write it down usually. That has been really helpful for me. Yeah.
If I had more money and foresight, I would do that. Instead, I have pencil. So
My first iPad I got for free because
Whoa.
my mom didn't want it anymore,
Whoa. Okay.
and then I just never could go back.
Yeah, that's valid.
Nice.
So.
Yeah.
So
Can I ask-
thanks, Mom.
Emmarie, can I ask you like an open question?
Oh, sure.
So,
Yes, please.
what was your like play-by-play memory of prelims, and did it like stack up to what you were expecting?
Like the actual test taking part?
Yeah, like the actual like oral examination.
Dude, my oral exam was crazy. I like spent so much time and effort putting together this like PowerPoint. And my advisor was like, your meeting is going to be long. Like, don't make it any longer than it has to be. Like, do a couple slides for each chapter. And like, the questions are just going to come. And, like I practiced a little bit, like how I was going to present this. And then I get maybe like 3 slides in and they're like, stop, I have a question. And then it just started. And it was like, I don't know, it was like maybe 2 or 3 questions per slide, and we just kept going, and then we like would stop maybe for like 10-15 minutes and just like go around and like ask a question. I think the other thing that like made it kind of interesting for me is that I think, I mentioned, so one of my committee members doesn't do anything related to sex chromosomes, and then 3 of the other committee members are all experts in sex chromosome evolution. And so it was like, the one committee member would ask like a really basic level question, and then they would all get so excited, and it would just like start flooding with like all these different like nuanced questions, and like it was pretty wild. I will say though that like I'm a bit of an emotional and anxious person, and so it was like cranking down to the 3rd hour, and I was like so exhausted and tired of just like going back and forth and answering questions and like feeling kind of like bad because I had to say I don't know a couple of times. Okay, and this is gonna sound really goofy, but I think that this is a really funny story, so just bear with me. So platypuses have 5 X chromosomes and 5 Y chromosomes, and they form this meiotic chain during meiosis in order for them to properly undergo meiosis, which is different than, placental mammal sex chromosomes. And one of my committee members was like, how do platypus sex chromosomes undergo meiosis? And I was like, oh, shoot. I don't know. Like, I know that they have 10, and I know that there's a chain, and And then one of them was like, well, how did they figure that out? And I was like, I don't remember. Like, I know that you told me, like, taught it in your class, but I don't know. And she was like, does PCR mean anything to you? And it was like, polymerase chain reaction. She was like, no, platypus chain reaction. And then like, I just started tearing up a little bit. And then they were all like, all right, break. She is really tired. I think we have asked enough questions. And so, yeah, I got an honorary platypus plushie a week after when I passed, because everyone in my lab thinks it's super funny that platypuses broke me, because our lab studies placental mammals, not monotremes, which are what platypuses are. So a question about a non-placental is what broke me. But that's really like the key thing that sticks out. Sorry.
That was an incredible response. Yeah.
What was the most memorable part of your, like...
Most memorable?
Yeah. Do you have a platypus question? Do you have a question that just broke you?
There was one question where my committee had asked me, I think I gave too many responses where I was like, oh, I'm gonna use this technique to do it. And they said, okay, imagine we lived in a world where there were no like transgenic techniques and you couldn't use them. And they're like, now answer this question. And I answered it. And then the question after that, I don't think anyone told me that we were going off of the no transgenesis allowed. And then they asked me a question that definitely required it. So they were asking me like the most basic question. They're like, how would you test to see if this pathway is interfering in, you know, like a microinjection thing? And I was like, oh, it'd be so easy if I could just say, yeah, you just insert this gene in the Pee-wee cluster and then, you know, you'll see if it interferes. But I was thinking that it wasn't allowed. So they watched me like almost have a meltdown because I thought I couldn't, you know, answer it in a certain way. And at some point they're like, okay, we're going to give you a lifeline and it involves transgenesis. And I was like, so
phone a friend, Dr. Adelman.
Yeah, it was.
Please help.
They were actually laughing at one point because I think they saw my head like steaming a little bit, and they were like, "This poor man." But honestly, I have to say there weren't too many crazy moments in my prelims. I actually like overhyped myself. I thought it was gonna be like 4, 5, 6 hours, and I rented the room or scheduled the room for that time, and I was in and out of there in like. I think it was just like 2.5 hours.
I wish.
It was like no.
You scheduled a room for 5 hours?
Yeah.
6.
Yeah.
I was
Should
ready
we
for
do
it.
that or?
I would say anticipate it being like 2.5 hours.
Okay.
Yeah.
All right.
Yeah, honestly, it was surprisingly no muss, no fuss, but I think it's because I had been a little nutty about preparing for it like in the previous months, that like when it actually came time to it, it was more like having a conversation and explaining my research.
Yeah.
That's
My committee
amazing.
members were so excited. They were like just wanted to talk about sex chromosomes and like evolution. And I was like, you guys are having way too much fun for the amount of stress that I'm feeling right now. Like, can we just like take it back? Yeah.
Sweet. Well, thank you for sharing all of your amazing advice. I think this episode is going to be very helpful for everyone who's trying to schedule their second committee meeting, such as the second years, and just for even the people who want to go to grad school to know what to expect in the middle of their studies here. So today's episode was hosted by Samantha Foster and Veronika Mojic and produced by Anthony Matarazzo. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, and X at Genetics Podcast. Thank you for listening, and we'll hope you'll tune in next time.